Good morning. This is Dr.Merina Paldas, Associate Professor of Department of Industrial Biotechnology. Today, I am going to explain about structure of DNA. So, in this topic, we are going to know about the introduction of the nucleic acids, definition of the DNA, location of DNA, and the different components of DNA. Then, the structure of DNA, different forms of DNA, and functions of DNA. So the nucleic acids are naturally occurring chemical compound that is the main information carrying molecules of the cell. And by directing the process of the protein synthesis, they determine the inherited characteristics of every living things. Although the name nucleic acid suggests their location in the nuclei of the cells, certain of them are, however, also present in the cytoplasm. So the nucleic acids are the hereditary determinants of living organisms. They are the macromolecules present in most living cells, either in the free state or bound to proteins as nucleoprotein. So we know that when the DNA will be bounding with the nucleoprotein, that will be the stable form of the DNA inside the nucleus. Because DNA, the length of the DNA is more than the radius of the cells, radius of the nucleus. So it has to be coiled with the help of the nucleoprotein, and it will be coiled in, packaged into the nucleus. So like the proteins, the nucleic acids are biopolymers of high molecular weight with the mononucleotide as their repeating unit. So the nucleotides are the monomers which is making the polymers of nucleic acid. So just how it is the amino acid are the repeating units of the protein. So there are two kinds of nucleic acid. One is a deoxyribonucleic acid. In a short form, we are telling as a DNA. And another one is ribonucleic acid, that is RNA. Both type of nucleic acids are present in all plants and animals. So DNA is found mainly in the chromatin of the cell nucleus, whereas the most of the RNA, around 90%, is present in the cell cytoplasm and a little about around 10% in the nucleolus. So it may be added that extranuclear DNA also exists. It occurs for like a mitochondria, and in case of the plants, it will be in the chloroplast. So DNA is the master blueprint for life, and it constitutes the genetic material in all free living organisms and most viruses. But the RNA, if you see that, it is also a genetic material of certain viruses, but it is also found in all living cells, where it plays an important role in certain processes such as the making of proteins. So if you find out the main difference between the DNA and RNA, DNA will be passing on the information from one generation to the another generation, where the RNA will be involved mainly in the protein synthesis. So now we'll talk about the DNA. So DNA is the largest macromolecule of human body. So nearly every cell in the person body has the same DNA. DNA contains all the genetic information that makes you who you are, and every individual organism has unique DNA like a fingerprint. So the essential for all known forms of life carries the genetic instructions used in the growth, development, functioning and reproduction of all known living organisms and many viruses. So when the information what is related to the growth, related to the development, related to the reproduction, all will be passing through the DNA from one generation to the another generation. So these instructions are found inside the every cell, are passed down from their parents to their children. Most DNA molecules are double-stranded helices. So DNA is a biopolymer. It is consists of nucleotide as a monomeric unit, and it is the DNA near the ribonucleic acid. The term DNA was given by Zacharis. So DNA is a double-stranded helical structure in eukaryote and prokaryote, but in virus it may be double-stranded or single-stranded, and it represents as a monoparotite or multiparotite. In eukaryotes, DNA presented in nucleus surrounded by the nuclear membrane. So that is a main difference between the prokaryotes and the eukaryotes. In case of the prokaryotes, DNA presented in nucleoid region of cytoplasm without nuclear membrane. In virus, DNA is presented in the core of virus surrounded by protein layer that is called capsid. So now we'll see the definition of DNA. So DNA or deoxyribonucleic acid may be defined as polymer of deoxyribonucleotides, which are connected by 3' 5' phosphodiester bond. It is the hereditary material in humans and almost all other organisms. So if you find the location where we can find out the DNA, most DNA is located in the cell nucleus where it is called nuclear DNA. Okay, but the small amount of DNA can also be found in the mitochondria where it is called mitochondrial DNA or empty DNA. So for a short note here, the whatever the DNA is coming from the parents to the children, like so from the father, whatever the nuclear DNA, it is coming from father and mother, but in the mitochondrial DNA, it is only come from the mother. So now we see the composition of DNA. So DNA molecules are double-stranded helices, and each molecule of DNA consists of two long biopolymer made of simple units that is called nucleotide. So there will be two polynucleotide chain. Okay, so that two polynucleotide chain is called as a two biopolymers. Okay, and they are that made of the single monomer unit that is called the nucleotide. Each nucleotide molecule contains a phosphate group, a sugar, and nitrogen base. So now we'll see the individual one. And if you see the sugar, it will be pentose sugar. That means it is a 5-carbon sugar. So it contains phosphorus in the form of phosphate, and this phosphate will be there in negative charge. So because of that, DNA is a negatively charged molecule. These nitrogen bases are like adenine, guanine, thymine, and cytosine. So it will be short form is denoted as A, G, T, C respectively. The order of these bases is what determines the DNA's instruction or genetic code. So they will be in different form, different sequence. That sequence only will give the particular information. So here I have shown with a picture in a very clear way. So the nucleic acids are polymers, right? So this polymers definitely made of some monomers. So that monomers name is called as a nucleotide. So if you see one nucleotide, it is having nitrogenous bases. So there are two types of nitrogenous bases. One is a purine, another is pyrimidine. And it will be there one sugar, that is a 5-carbon sugar. There are two types of sugar. One is a ribose, one is a deoxyribose. So in case of DNA, that sugar will be deoxyribose, and in case of the RNA it will be ribose. So if you see the picture, so that both cases it will be ribose. So but in case of the DNA, that will be deoxyribose. That means in the carbon 2' position there will be no oxygen, right? So that's what it is called deoxy. Okay, so deoxyribose. And if you see, there will be one phosphate group, and so this so nucleotide is made of nitrogenous base, sugar, and phosphate. So if you take only the nitrogenous base and the sugar, that is called nucleoside. Okay, so the nucleotide equal to nucleoside plus phosphate. So here you have given a tabular form where we can see the components of RNA and DNA clearly. So we know that there is a common part is acid, that is a phosphoric acid in both sides, and there will be one pentose sugar, but there will be not same sugar, right? One will be the RNA, it will be ribose, whereas then the DNA, 2 deoxyribose. So we have seen that in the 2 position there is a removal of oxygen, so that is called deoxyribose. Now we'll talk about the nitrogenous base. So if you see the nitrogenous base, purines will be the same for DNA and RNA. So that is adenine and guanine. But in case of the pyrimidines, we can see in case of RNA, it is cytosine and uracil, and in case of DNA, it is cytosine and thymine. So if you take the hydrolytic products of nucleic acid, like if you take the nucleic acid and if you go for the hydrolysis, okay. So here we are given the 3 conditions. So first condition is we have taken the aqueous ammonia at around 115 degrees centigrade, or barium hydroxide. If we take that, it will be degraded to, the nucleic acid is a polymer, right? So it will be degraded to nucleotides. Even if you take the nuclease enzyme, that also it will break down to nucleotides. Now, if you take the nucleotide and if you boil that nucleotides around 175 degrees centigrade in aqueous ammonia, it will break down to nucleoside and phosphoric acid. Okay, and the same nucleoside we can get it from nucleic acid also. That time what you have to do, we have to take the nucleic acid in magnesium oxide in solution, and you have to heat it. So after that, this nucleoside we can add the inorganic acid, and this inorganic acid will break the bond between the sugar and the bases, like it may be a purine base or it may be a pyrimidine base. So we can get all the components separately. So sugar, purines, and pyrimidine. Okay, and if you see that nucleic acid will be higher molecular weight compared to that nucleotide will be compared to less, Then it will be nucleoside that will be compared to less, and from the nucleoside we will be getting separately sugar, purines and pyrimidine. So this is the decreasing order of molecular weight. So now we'll talk about the individual component. So first we'll talk about the phosphoric acid. So the molecular formula of phosphoric acid, we know that everyone H3PO4. So it contains a phosphate group, is just a pentavalent phosphorus atom. Pentavalent means there will be 5 valency of the phosphorus atom, and which will be bound with the 4 divalent oxygen atom. And it has one important role, like I told you that it will be carrying the negative charge. For that it will be performing so many important functions. So along with the sugars and bases, it make up the nucleic acid. So next we'll talk about the pentose sugar. So the 2 pentose sugar means that is a 5 carbon sugar. So 5 carbon sugar means we know that in case of the Haworth formula, it is taking the furanose ring. The 2 types of nucleic acid are distinguished primarily on the basis of 5 carbon keto sugar or pentose which they possess. One possesses D2-deoxyribose. Hence, the name of the nucleic acid is deoxyribonucleic acid. While the other contain D-ribose. Okay, so hence that ribose containing nucleic name is ribonucleic acid. That is a RNA. So an important property of the pentose is their capacity to form esters with phosphoric acid through phosphodiester bond. So it is a type of covalent bond, and the linkage between the 3' carbon atom of one sugar molecule and the 5' carbon atom of the another sugar molecule, there is a deoxyribose in DNA and ribose in RNA, it make up the backbone strands of nucleic acid. This bond, in fact, is an integral part of the structure of nucleic acid. So when you'll be seeing the different, we'll be seeing the different level of the structure of DNA, we'll be seeing that the 3' 5' that phosphodiester bond, that is the main part of this structure. So now we can see in a picture that is that first and second ester linkage between, and it will be the we are shown here between the two sugars. So 3' OH of the first sugar making the 5' CH2OH bond. So as it is having the 2 oxygen, so that's what, and with the help of phosphorus, so it is called phosphodiester. Okay. So the one nucleotides get connected with another nucleotide by 3', 5' phosphodiester bond. The 5' end of newly added nucleotide makes connection with the 3' end of existing nucleotide. So that is called the primer to elongate the chain. So like that only, that all the nucleotide will be adding and they will be getting a big chain of polynucleotide. So nucleic acid chain always elongate by polymerization from 5' end to 3' end. So DNA will be the first one chain, if you consider, it will be always increasing from 5' end to 3' end. After completion of the synthesis, on one end side 5' end will be always remain free. On the other side, 3' end will be always remain free. So if you see that the 3' end will be always free with one phosphate group, and where the 3' end, it will be always free with the OH group, hydroxyl group. So this one will give it will provide the polarity to the nucleic acid chain with 2 distinct ends. It is a tradition to read the base sequence of nucleic acid chain from 5' end to 3' end. So now we'll talk about the nitrogenous bases. So there are 2 types of nitrogenous bases are found in all nucleic acid, either it is a DNA or RNA. So the nitrogenous bases are derivatives of pyrimidines and purines. So pyrimidine bases means it will be made of only one 6-member ring, and under this category examples are uracil, thymine and cytosine. Where the purine bases, this is made of a 6-member ring and it is fused with another 5-member ring. So example under this category is adenine and guanine. So pyrimidine derivative, these are all derived from their parent heterocyclic compound pyrimidine, which contain a 6-member ring with 2 nitrogen atom and 3 double bonds. So this is a basic structure, and the numbering will start from the bottom end, and it will be from the right hand side 1, 2, 3, 4, 5, 6. And the melting point of this pyrimidine generally around 22 degrees centigrade, and around boiling point will be 120.5 degrees centigrade. So this is the structure of that uracil, thymine and cytosine. So uracil is C4H4O2N2. It will be found only in RNA, and molecular weight around 112.10 daltons, and melting point 338 degrees centigrade. Where the thymine is a molecular formula C5 H6O2N2. It will be found in the DNA only, and molecular weight around 126.13 daltons, melting point around 335 degrees centigrade. Where the cytosine is molecular formula C5H4ON3, and it will found in both DNA and RNA, and it is molecular weight around 111.12 daltons, and melting point will be around 320 to 325 degrees centigrade. So if you see that here will be the carbonyl group will be there, NH2 group will be there. This will help them to make the bond with the another polynucleotide chain. So now we'll talk about the purine derivative. So these are all derived from their parent compound that is called purine, which contain a 6-membered pyrimidine ring that is fused to the 5-membered imidazole ring, and it is related to uric acid. And it has a melting point around 260 degrees centigrade. So here we can see that left hand side, the nitrogen, from here only the count will start 1, and it will be then from the anti-clockwise like 1, 2, 3, 4, and then it will finish the 6-membered ring, then it will go to the 5-membered ring. So now the example of this under this category is adenine and guanine. Adenine will be the molecular formula C5H5N5, and it will be found in both DNA and RNA with the molecular weight around 135.15 daltons and melting point around 360 to 365 degrees centigrade. Where the guanine around the formula is C5H5ON5, it is also found in both the DNA and RNA with molecular weight 151.15 daltons and melting point around 360 degrees centigrade. So here that the bond between the that NH2 or the carbonyl group, it will help to join with the another polynucleotide chain. So now we'll talk about the nucleoside. The nucleosides are the compounds in which nitrogenous bases, that means either pyrimidines or purines, are conjugated to the pentose sugar. So pentose sugar may be a ribose or deoxyribose by a beta glycosidic linkage. So when the pentose sugar and nitrogenous base will be attaching, the bonding between them is glycosidic linkage. So the beta glycosidic linkage involves the C1 prime of sugar and the hydrogen atom of N9 in case of the purine, and in case of the pyrimidine it will be N1, thus eliminating a molecule of water. So with the elimination of the water, in case of the purine it will be C1 prime and N9, in case of the pyrimidine C1 prime and N1. So this is the structure of this nucleoside where the sugar and base. So we can see here that that C1 prime and the nitrogen N9 is making base. It is for purines, and in case of the pyrimidine that will be the N1. So what will be the name protocol? So in case of the ribonucleosides, adenine and ribose, so base and sugar. So it will be adenosine. Guanine plus ribose, it will be guanosine. Uracil plus ribose, it will be uridine. Cytosine plus ribose, cytidine. Hypoxanthine plus ribose, inosine. It is a derivative of purines. Then xanthine plus ribose, xanthosine. Where it will come for the deoxyribonucleosides, it will be adenine plus deoxyribose sugar. So it will be the same name, but the suffix will be deoxy. So deoxyadenosine, and it will be denoting as a small d. Okay, so deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Okay, but deoxyuridine will be not there because uracil will be not there in case of DNA. So now we'll talk about the nucleotides. Nucleotides are the phosphoric acid ester of nucleoside. So I told you that there will be phosphodiester bond, and these occur either in the free form or as subunit in nucleic acid. In the ribose moiety of a ribonucleoside, phosphorylation is possible only at 3 positions. One is C2', C3', and C'. So since C1' and C4' are involved in the furanose ring formation. In the deoxyribose component of a 2' deoxyribonucleoside, only 2 positions are available. That is C3' and C5'. Okay. So in case of RNA, 3 positions, but in case of DNA, only we have seen 2 positions are available for phosphorylation. Since in this sugar, C1' and C4' are involved in the furanose ring, and C2' does not bear a hydroxyl group. So that's why it will not participate. So here you see the nucleotide. So sugar and base already it is attached. In that what? Only the phosphate group attached. So phosphoester bond, and this is I'm talking about the only one nucleotide. So when the another nucleotide will come, that time only phosphodiester will be there. So the phosphoester bond, so 1, 2, and 3 phosphate group, and that's what at the 3' at the 5' hydroxyl group. Okay. So now if you see the name pattern of the nucleotide, so adenosine already it was there when it was added the sugar and base. So in that phosphate added, and the name is adenosine monophosphate. So it will be short form is AMP, and we can tell also adenylic acid. Similarly, with the guanosine, it will be guanosine monophosphate or guanylic acid. Cytidine plus phosphate, cytidine monophosphate or cytidylic acid, CMP. Where the uridine and PI, it will be uridine monophosphate, UMP or uridylic acid. Inosine plus phosphate, it will be inosine monophosphate or IMP or inosinic acid. When we'll talk about the deoxyribonucleotides, here the D-adenosine plus PI, same thing, the adenylate acid, it will be suffixed with D, like deoxyadenylate acid, deoxyguanylic acid, deoxycytidylic acid, or deoxythymidylate acid. So DMP, DGMP, DCMP, and DTMP. So now we'll come to the structure of DNA. So till now we have seen what are the components of DNA. Now we'll see the structure of DNA. So before going to the structure of DNA, so I told you that there will be two polynucleotide chain. So this polynucleotide chain, how they will be their arrangement. So the base composition that has given the scientist Chargos. So he has framed some the rule that is called Chargos Rule. So based on the Chargos Rule, DNA base composition will be, they'll follow this 5 or 6 rules are there. So the first rule is that the sum of the purines is equal to the sum of the pyrimidine. So it is for one DNA. Okay. So if you see that, we can say that A plus G equal to T plus C. That means total how many purines are there? Pu equal to Py. Where the ratio of the adenine to thymine is also 1. That is also another rule. So that means A by T equal to 1. Where the ratio of the guanine to the cytosine is also 1. That is G by C equal to 1. Bases with the 6 amino groups are equal to the bases with 6 keto or maybe the hydroxyl groups, because keto and hydroxyl, we know that there will be ketone tautomerism. So in this context, we can see that A plus G equal to G plus T. And the ratio of A plus T by G plus C, it is also called dissymmetry ratio. It will vary greatly from one species to another species. And it is also characteristics for a particular species. Chargos data suggests that A is always paired with T and G is always paired with C. So that's what the base composition rule will follow all these 6. So there are 3 levels. So first level is a primary structure. So primary structure of the DNA refers to the phosphodiester backbone. So here last slides and all, we have seen that the 3', 5' phosphodiester bonds. Okay. So that is the main part of this primary structure. In nucleic acid strands, nucleotides are linked by phosphodiester bond. The bond is formed between the 5' phosphate group of one nucleotide and 3 hydroxyl group of adjacent nucleotide. A single nucleic acid strand formed by phosphodiester bond has 2 termini, namely 5' end with phosphate group and 3' end with hydroxyl group. So like that only the one strand will be there for the primary structure. So now we'll talk about the secondary structure, and secondary structure will be explaining about the Watson-Crick model, and they have proposed around 1953 the double helical DNA model. So this is also called as a B form. So DNA molecule consists of 2 helical polynucleotide chains, which are called helically around or wrapped around a common axis. So the double helix is generally right-handed. That is, it turns clockwise looking along the helix axis. The side of the ladder are made up of alternating molecules of phosphate and deoxyribose sugar and purines and pyrimides on the inside of the helix. So now we have seen that the sugar phosphate will be the outer part and that the bases will be in the inside the part. The 2 strands run anti-parallel. So here 2 polynucleotide chains will be there. So we have seen that how the primary structure, it is running from the 5' to 3' side. So here as the 2 chains will be there, they will be anti-parallel. That means one strand has phosphodiester linkage in the 3' to 5' direction, while the other strands will be there phosphodiester linkage in the 5' to 3' direction. So the phosphate and deoxyribose units are found on the periphery of the helix, whereas the purines and pyrimidine bases occur in the center. The planes of the bases are perpendicular to the helix axis. The planes of the sugars are almost at the right angles to those of the bases. If you see the diameter, it will be around 20 angstrom, and the bases are 3.4 angstrom apart along the helix axis. Therefore, the helical structure repeats after 10 residues on each chain. That means an interval of 34 angstrom. We can say, in other words, that each turn of the helix contain around 10 nucleotide residues. The two helices are wound in such a way that to produce two interchain spacing or groups. So there will be one major group and there will be one minor group. So major group, we can say another one is wide group. And if you see that, there will be around width around 12 angstrom and depth will be around 8.5 angstrom. Where the minor group, that will be around width will be 6 angstrom and depth will be 7.5 angstrom. So the bases makeup of the stairs of the ladder are attracted by a weak chemical bonds that is called hydrogen bonds. So the two, the polynucleotide chain, they are twisted, they are in helical. This helical, the position making stable by the hydrogen bond. So we know that hydrogen bond is weak bond, but still this is a stable structure because there are a number of hydrogen bonds are more. So a chemical hydrogen bond, if we define it, is a chemical bond in which a hydrogen atom of one molecule is attracted to an electronegative atom of the another molecule. So it may be a nitrogen, it may be a oxygen, it may be a fluorine atom. So the two strands are held together by hydrogen bonds between specific base pairs of purines and pyrimidines. So as per the Chargos Rule, we have seen that the hydrogen bonds between the purines and pyrimidines are such that adenine can only bond to thymine. And how many hydrogen? There will be 2 hydrogen bonds. And guanine can bond only to the cytosine by 3 hydrogen bonds. The specificity of the kind of hydrogen bond that can be formed assures that for every adenine in one chain, there will be thymine in the other of every other chain. And for every guanine in one chain, there will be cytosine in the other chain. So thus, the two chains are complementary to each other. The two hydrogen bonds connect to T to A. See here in the picture also you can see that, and the 3 hydrogen bonds connect to G to C. The individual hydrogen bonds are weak in nature, but as in the case of the proteins, a large number of them involved in the DNA molecules that will confer the stability to it. It is now thought that the stability of the DNA molecule is primarily a consequence of van der Waals forces between the planes of stacked bases. So this also may be giving additional force to stable the structure. So now you can see that the sequence of the bases along of the polynucleotide chain is not restricted in any way. The precise sequence of bases carries the genetic information. As a corollary, the entire structure of a DNA molecule resembles a winding staircase, with the sugar and the phosphate molecules of the nucleotides forming the railing and the linked nitrogen base pairs as AT and GC forming the steps. The double helix is a spiral right-handed, and that is each of the nucleotide chain turned right. This can be verified if we look at the segment where the threads move upward and eventually turn right. If the two strands rotate clockwise, it is said that the double helix is right-handed, and if they turn towards left, it is called left-handed. This form may appear in helicals alternately because of the conformational changes in DNA. But the most common conformation adopted by the DNA double helix is right-handed, turning every couple of bases in the previous approximately around 36 degrees centigrade. So now we'll see the tertiary structure. It is a supercoiled DNA. Chromosomes and chromatin are the 3 types of tertiary structure of DNA. DNA can be twisted, coiled into compact structure, which resembles a coiled telephone wire. Supercoiling may be right-handed or left-handed. So now we'll see the different forms of DNA. So here we can see that aDNA, it is one of the many possible double helical structure of DNA. It is most active along with other forms. Helix was right-handed sense, shorter, more compact helical structure. It occurs only in dehydrated samples of DNA, such as used in crystallographic experiments. So if you see the structure of aDNA, it is a fairly similar to the bDNA. Slight increase in the number of base pair rotation resulting in tighter rotation angle and smaller rise per turn. Deep major group and a shallow minor group. It is favored conformation at low water concentration in a solution with higher salt concentration or with alcohol added. The DNA structure may change to an A form, which is still right-handed, but every 2.3 nanometer makes a turn, and there are 11 bases pair per turn. Now we'll see the bDNA. So this is the bDNA we have seen in the normal double helical structure, which is called as a Watson-Crick model. It will be first identified in fiber at 92% relative humidity. It will be helical since right-handed. Base pair almost perpendicular to the helix axis, around 3 angstrom apart, and one turn of the helix around 36 angstrom, that is approximately 10.4 base pairs, and minor group around 12 angstrom across, and major group around 22 angstrom across. Now the last form, that is the zDNA. It is one of the many possible double helical structure of DNA. Helix has a left-handed sense. It is the most active double helical structure. zDNA was first discovered in 1979, and certain proteins bind very strongly to the zDNA. So here we are given the tabulated form of the different forms of DNA. So here we can see that aDNA right-handed, even bDNA right-handed, but the zDNA left-handed. Basis pair per turn, it will be for aDNA 11, bDNA 10.4, and zDNA 12. Rise per base pair along the axis around for aDNA 0.23 nanometer, bDNA 0.34 nanometer, zDNA 0.38 nanometer. Diameter, if you see this, for the aDNA 2.55 nanometer, bDNA 2.37 nanometer, and zDNA 1.84 nanometer. Pitch for aDNA 2.46 nanometer, 3.4 nanometer for bDNA, 4.56 nanometer for zDNA. Major group present for aDNA and bDNA, but it is absent in zDNA. Minor group also present aDNA and bDNA, but there will be deep cleft will be there only zDNA. So now we'll see one important properties of this DNA. It is called hyperchromic effect. It is the increase of absorbance of optical density of a material. The most famous example in the hyperchromicity of DNA that occurs when the DNA duplex is denatured. The UV absorption is increased when the two single DNA strands are being separated, either by heat or by addition of denaturant agent, or increasing the pH level. The opposite, a decrease of absorbance, is called hypochromicity. The functions of DNA, if you see that, scientists have discovered that the certain proteins bind strongly to the zDNA, suggesting that zDNA plays an important role biological in protection against the viral cell. The main role of the DNA in the cell is a long-term storage of information and passing from generation to generation.